MacConkey Agar. It's a selective differential medium, because it contains bile salt and crystal violet to inhibit the gram-positives, and lactose to differentiate bacteria into lactose fermenter, LF, which is form, red colonies, and non-lactose fermenter, NLF, produce, white/pale pink/yellow colonies. pH indicator of MacConkey medium is a neutral red. What are pH indicators? pH indicators. An acid-base indicator is often added to differential media to detect changes in hydrogen and hydrogen ion concentration during the growth of an organism. Generally, this change can be due to different metabolic processes, such as fermentation, utilization of substrates, deamination, hydrolysis, oxidation, and nitrogen fixation. How does crystal violet inhibit the growth of gram-positive bacteria? 1. Cell wall interaction. Crystal violet, a positively charged dye, binds to the negatively charged components of the thick peptidoglycan layer in gram-positive bacteria, such as teichoic acids due to electrostatic interactions. This binding prevents proper cross-linking of the peptidoglycan, weakening the cell wall and increasing membrane permeability. Consequently, the bacteria experience osmotic imbalance, leading to cell lysis and death. 2. Inhibition of cellular functions. Once inside the bacterial cell, crystal violet interacts with intracellular components, particularly enzymes and proteins that are critical for cellular metabolism and function. It interferes with DNA replication by binding to DNA strands, preventing proper transcription and subsequent protein synthesis. The dye can also disrupt the functioning of enzymes involved in metabolic pathways, halting essential processes such as energy production, which further inhibits bacterial growth and survival. 3. Disruption of membrane function. Crystal violet can also affect bacterial cell membranes. Gram-positive bacteria, which have a more exposed membrane system due to their thicker peptidoglycan layer, the dye can alter membrane permeability and function, impairing nutrient uptake and waste removal, and ultimately affecting cell viability. Gram-negative bacteria, on the other hand, possess an outer membrane made up of lipopolysaccharides, which reduces the dye's ability to interact with their cell wall, making them more resistant to crystal violet's effects. How do bile salts inhibit the growth of gram-positive bacteria? Bile salts in MacConkey Agar inhibit the growth of gram-positive bacteria by disrupting their cell membranes. They act as detergents, damaging the lipid bilayers of gram-positive cell walls, leading to cell lysis. A modified MacConkey Agar. Sorbitol MacConkey, used to identify Escherichia coli 0157. What is Escherichia coli 0157? Is a pathogenic strain of E. coli that causes severe foodborne illnesses. It is a Shiga toxin-producing E. coli, STEC, meaning it produces toxins that can lead to serious health complications such as hemorrhagic colitis, bloody diarrhea, and hemolytic uremic syndrome, HUS, a condition that can cause kidney failure, especially in young children and the elderly. What are the differences and similarities between Sorbitol MacConkey Agar, SMAC, and standard MacConkey Agar? Sorbitol MacConkey Agar replaces lactose with sorbitol. E. coli 0157 cannot ferment sorbitol. In Sorbitol MacConkey Agar, non-pathogenic E. coli strains will ferment sorbitol and appear pink, while E. coli 0157 will not ferment sorbitol, producing colorless colonies. Both agars contain bile salts and crystal violet to inhibit the growth of gram-positive organisms, and pH indicator is neutral red. Question and answers. Question 1. What is MacConkey Agar used for? MacConkey Agar is a selective and differential medium, primarily used to isolate and differentiate gram-negative bacteria, based on their ability to ferment lactose. It inhibits the growth of most gram-positive bacteria due to the presence of bile salts and crystal violet. Question 2. How does MacConkey agar differentiate bacteria? MacConkey agar differentiates bacteria by their ability to ferment lactose. Bacteria that ferment lactose produce acid, turning the pH indicator neutral red, pink or red, while non-lactose fermenters form colorless or pale colonies. Question 3. Why are gram-positive bacteria inhibited on MacConkey agar? Bile salts and crystal violet in MacConkey agar inhibit the growth of gram-positive bacteria by disrupting their cell membranes and interfering with cell wall integrity, making it selective for gram-negative bacteria. Question 4. Can gram-positive bacteria grow on MacConkey agar? Normally, gram-positive bacteria do not grow on MacConkey agar. However, rare cases of mutant strains, like Streptococcus pneumoniae, have been reported to grow under specific conditions. Question 5. What color do lactose fermenters appear on MacConkey agar? Lactose fermenters, such as Escherichia coli, appear pink or red due to acid production that changes the pH indicator's color. Question 6. What are the key components of MacConkey agar? The key components of MacConkey agar include bile salts, crystal violet, lactose, neutral red, pH indicator, and peptones. Bile salts and crystal violet inhibit gram-positive bacteria, while lactose and neutral red help differentiate lactose fermenters from non-fermenters. Question 7. Can MacConkey agar be modified for specific bacterial studies? Yes, MacConkey agar can be modified by altering its components, such as adding sorbitol for distinguishing E. coli 0157. Thank you and follow for more.